Welcome back to Bioinformatics with BB channel. My name is Babajan Bananganapalli. I'm an associate professor in bioinformatics. In this video, we are going to learn about how we can design a primer by using NCBI Primer BLAST. PCR is most commonly used method to amplify the DNA of interest in many fields such as biomedical research, diagnostic testing, and forensic science. The outcome of the PCR product is majorly depends upon the three factors. One is the preparation of the template DNA, and the second one is reaction conditions, and third one is designing of a good pair of a primers. If you take the general requirement for designing of a primer, we have to follow five different rules. One is the length of the primer should be always in between the 18 to 24 base pair in length, and the GC content should be balanced. It means it should be around 40 to 60% GC content should be there, and melting temperature you have to maintain in between the 50 to 60 degrees centigrade. And we need to avoid the self complementary. It means we need to avoid the hairpin formation. If if the 3 prime end of the primer is complementary to the 5 prime end of the primer, it is going to form a hairpin leg structure. Okay, we have to avoid the self complementary primers and also the cross primer formation. If the any region of the forward or reverse primer is complementary to each other, they are going to form a dimer like structure. Okay, these are the five things we have to remember before we go for the designing of a proper primer. So luckily for us, we need not to remember all these rules. Nowadays we have so many softwares are already available. Just you have to provide a sequence to the softwares or tools. They are going to give a predicted primers for you. So always we have to remember, we have to cross check that whether these primers are covering our product or not, whether they are following the rules of primer designing or not. These things we have to cross check each and every set for each and every set of the primer that designed by the software. So you can see here so many software nowadays, so many tools or softwares are available for designing of a primer. So out of them, the primer 3 is a most used or most commonly used tool for designing of a primers. Okay, so here the major disadvantage with this other tools is they are not target specific. It means, so whatever the primer they will generate, they are not going to check whether this primer has been in any homologous region with other genes or not. So to avoid these circumstances, we have a NCBI Primer BLAST. So this NCBI Primer BLAST will specifically use this primer 3. Okay, it is embedded with the primer 3 along with the NCBI BLAST. So what it will do is, not only it will generate a primers for you, and also it will give a target specific region. Here we are going to take example of MCC gene exon 2. It's human colorectal methane cancer protein exon 2. So we are going to design a primer for this MCC gene. So we always need to remember the primer synthesis of the DNA. It majorly depends upon the various factor. So it depends upon the sequence of the DNA, and also it depends upon the type of reactions, and also it's also majorly influenced by the type of instruments we are using. So nowadays most of the researchers or most of the research labs, they are using the ABI 3500 instrument, which is very commonly used instrument for the Sanger sequencing. So here what we will do is we are going to retrieve the nucleotide FASTA sequence from NCBI. So my aim is I want to cover the whole exon. Okay, so I want to cover the whole exon. Let me see what is the length of the exon. As I told you, the primer maximum capacity will be around 1000 base pair. Always you have to, if it is more than 1000 base pair, what we need to do is we have to split your exon or your target region. Okay, you have to split your target region into two, then you can design two different primers for that one. So if it is more than 2, for example, just take the MCC gene, it consists of nearly 14 exons are there. If you want to cover all the 14 exon, my advice is don't go for the Sanger sequencing. It's always better to go for the exome sequencing, which is much cheaper than the Sanger sequence, where it will cover all the exons human DNA sequence. I want to cover the almost all exon 2. So I'm going to retrieve the sequence from NCBI in FASTA format. I'm going to design a primer by using NCBI Primer BLAST. So I want to check the specific target site, whether it's mapping with the same target site or either mapping with the other target sites. I'm going to cross check that one. Finally, I'm going to validate this one, whether these primers are following the 5 rules or not, and also I'm going to look onto the different properties of the primers by using the Primer Start program. Okay, so let us start. Okay, this is the second exon FASTA sequence. Okay, so here copy the sequence and paste in a WordPad. So here this is the exon 4 sequence, exon 2 sequence, and just copying paste this sequence here. So now I want to design a primer for this sequence. If I design a primer for this sequence, I'm going to miss some of the regions. Okay, at the starting, always you have to remember your Sanger sequencing. When the start of the primer, always you will find the shutterings. Okay, so the mismatch shutterings always you will find. Once it's crossed to the 50 to 60 base pair, then only you will find a very smooth reading or smooth DNA synthesis. If I design a primer in an exon region, always I have a chance to miss some of the very key residues at the 5' and 3' end. So to avoid this region, to avoid this circumstances, I can go to the intronic regions. Okay, I can go to the intronic region. If you want to go to intronic region, if you want to go to sync region, you have to extend the sequence. So we can extend this sequence very easily in NCBI. So for example, I'm going to extend this sequence. So I'm going to add 400 more to it. That means the sequence is going to start from 619. Instead of 619, it's going to start from 419. Okay, so the 5' region 419, and here the 3' region I'm going to add 100 base pair mode. So the 5' region I added 500 base 200 base extra to the sequence, and here the 3' end I added 100 base pair extra. Okay, if I click on the FASTA, now we can see this added sequence you will find. So here copy the sequence, again paste here. Okay, so let us find where exactly the exam has been started. Copy. Okay, so here paste here. You can see here the exam has been started here. Okay, so exam has started here. Let us see ending region. So here this is the end region. So highlight everything, the whole exon. This might be helpful for us when we are starting the primer design. Okay, so now we have the sequence. We have the intronic regions are there, exon region, the 5' intronic region and the 3' intronic region has been added to the exon region. The copy the sequence. Now it's time to start NCBI Primer BLAST. Go to NCBI Primer BLAST. So you can click on directly pick primer here, or you just open the home page, paste the sequence here. Okay, so range here, in this case we can add a range. If you want to start a primer, if you want to have a primer in the specific region, you can give this region here. Okay, so for example in our case I'm going to give this region. I just want to start my primer only in this region. Okay, so only this region I want to start from 1 to, from first base pair to, I don't want to cover my, I don't want to touch my target region. Okay, so here 1 to 173, 1 to 173 region. Okay, okay, so for the reverse one we have to select starting from this region 4, we're going to select from 459. Okay, so here add 459 to the end region. Okay, so let's see the end region also here. So the end one is 545, so 545. Okay, so here this is the range I want a primer only this region. I don't want to give other region. So why means, as I told you, so my target is our primer need to cover all the exon. Okay, if you just keep this one as a default, even it will search here also. Okay, I don't want to search this, I don't want to search the primer 3 in my target region. Okay, so if you want to check the total primers for your sequence, you can clear this field, you can search by default. The next one, the primer parameter, if you already know the parameter, sorry, if you already know the primer, you have to paste this sequence, forward and reverse sequence here. It will show you where exactly your primer has been there in the sequence. And product size also, you can if you want, you can mention the product size or how much product size you want. So I want by default it will cover, my primer will cover approximately 300 to 400 base pairs for my sequence. Okay, so it minimum of for 72, maximum of 1000 base pair it will cover. So primer returns how many primer sets you want. We just by default we are going to keep it 10. Keep remaining everything as a default parameters. You don't change anything. So except here you have to select the database. If your sequence is exon 1 coding 1, then you have to select the reference sequence database. If your sequence is including the intronic region, then you have to select the non-redundant database. So in our case we included both the exons and introns. Click on NR and click on get primer. So one good thing with this NCBI Primer BLAST is it will search first. It will search your sequence in a database. It will map with the known or unknown gene. For example, now we pasted a sequence, it identified that 2 regions are there. So one is clone region and another one is the known gene region. So you have to select always known gene region. So here this is our gene, MCC gene, and click on submit. Now we got the primer list. So you can see here the first primer set. So here it is forward primer and this is a reverse primer. It is almost giving the 490 base pair product size for us. Okay, let us find out where exactly it is there in our sequence. Okay, copy the sequence here, paste here. You can see here this is located this region. Okay, so here color them like this, and the reverse one, so you don't find the reverse one. Why means it is in reverse complementary sequence. Okay, so you cannot find this sequence, so you have to convert this sequence into the forward sequence. So here you have to use the tool reverse complement. Type reverse complement in Google, like reverse complement. You will find this first one. Okay, so paste your sequence here, and you get this sequence here in this region. So it is somewhere here. Okay, it is GCC TCA TCA, right? TCA. Okay, so this is a reverse sequence. So now we have a reverse sequence. So this is a reverse sequence and forward sequence. So here the forward sequence. Okay, so the major disadvantage with the NCBI Primer BLAST is, so most of the time it's going to give a false positive primers for you. Okay, so always we have to cross check all this primer with one more tool, which is a very good tool. It not only give you a properties for you, not only it will assess the primer quality for you, and also it will give a how we can set up your PCR reaction. So for this we have to go to the tool called as a Primer Start. Okay, so click here the PCR Primer Start. So this is a very good tool where even they have the offline version is also available here, where you can download this version in your system. You can work with this one. So here you simply need to paste the forward sequence and the reverse sequence. Okay, so I'm going to copy this forward sequence here, the reverse sequence of reverse sequence. Okay, so copy paste here the reverse sequence, click on submit. So now it will show you information for you. Now you can see here, so we got an error with the first primer, forward primer. So the warning is GC is less than 40. Okay, don't worry about this warning. If you find the warning, just ignore it. As I told you, it should be covered to 50 to 60 degrees TM. It will cover. Okay, so 58 is it's just warning, it's negotiable. But if you got the warning anywhere, so anywhere, and this anywhere like the lens, single base pair run, self-funneling hairpin, you have to change the primer. Okay, so you can see here clearly the GC content is very low here, only less than 40%. So that's why we need to improve the GC content. Let us see where exactly it is there. So here, so the GC content is very less. What I will do is, I just want 20 base pair, I don't want more than that one. Okay, so I will remove this 3 base pair. I can start my primer from here. Okay, I can start my primer here. So I'm doing here my manual curation, so I'm not depending on the software is providing the false positive results for me, so where the GC content is very low in my primer. So what I will do is, so as my primer length is 24 base pair, as I told you, the length of the primer should be in between the 19 to 24 base pair. I'm going to remove last 3 base pair. So I'm going to start my primer starting from this region. Okay, so I'm going to use this region here. Let us see whether it's going to work or not. Copy this region and let's forward paste here and click on submit. Now we can see here the GC content has been covered. Now the GC percentage is very good for us. So like this, you have to use the manual curation sometime to pass your primer, okay, to get the very good primer. And you also always need to be very cautious when you are adding or shifting the frame. You cannot delete the residues in the middle of your primer, so you always have to remember, you have to delete whether in the 5' region or 3' region if you want to extend or if you want to base pair to it. Okay, you can do it. Even you can, if you want to cross check this one, you can do it in NCBI itself. If you want to get the course of your primer, okay, you have to paste this power primer here and paste the reverse primer. Okay, so here this is known one. Click on get primer. Now you can see this scores has been improved a lot. So it's 21 base pair in length. It has been starting from TM is pass, it's more than 50, and here GC content also is more than 40 is there. Self complementary score is less than 8. Okay, so in according to primer 3, if the self complementary score should be always less than 8, self 3 prime complementary score should be always less than 5. So let's copy this one. So this information is very useful for us when we are setting your PCR reaction. So paste this information. Okay, so even as I told you here in primer start, set up your PCR reaction by including all these parameters. Okay, so here that's the beauty of this primer start. Not only it will give you a primer quality scores, but also it will provide you this information for you. So, and one more thing you have to remember, when you're designing your primer, always you need to 100 base pair away from your target site. For example, as I told you here, my target site is this region. That's why I selected 100 base pair away from this region. So this is how we can design the primer by using the NCBI Primer BLAST and the Primer Start. If you like this video, please like, share and subscribe to our channel. Thank you so much for watching.